Sadly, the church is in decline today.
If truth does not exist, we will not be able to live and function as civilized individuals in a society.
Students in the public education system at large of our nation are left with the impression God's irrelevant because he's never mentioned.
We sort of have national Alzheimer's. Here we are, the freest country that the world has ever seen, with more individual freedom and opportunity to choose what you want to do with your life and where you want to go to church. We have that, and we forgot how we got here.
So we're going to answer the question, why all the fuss?
Despite ever-increasing efforts to undermine the authority and accuracy of God's Word, the Bible stands firm.
Science and the Bible are inextricably linked.
We see evidence of incredible design.
God has made it incredibly clear.
Men, listen to me today. God has a calling on your life. Kids need you. Women need you. Our country needs fathers and responsible husbands today.
55 out of 56 signers of the Declaration of Independence were Bible-believing, church-attending Christians.
We have the longest ongoing constitutional republic in the history of the world.
The Western world was successful because it was built upon biblical values.
All right, let me get my clicker turned on here. Okay, so I am ready to go. Is everyone wide awake? more or less. So the title of my talk is Monkey Business in the Chimp Genome, and you'll understand why I picked that here shortly. So, Charles Darwin was famous for this one book in particular, but he also published another book on human evolution, which I'll talk about. But The Origin of Species by Means of Natural Selection. But look at this other title here, the rest of the title, The Preservation of Favored Races in the Struggle for Life. And Charles Darwin was a racist, and he had a lot of friends that helped him promote those views. But Charles Darwin also published another book that was very influential as well, The Descent of Man. And in this book, he proposed that humans descended from apes, from an ape-like creature. And so that's really, while there were some ideas floating around about that before, Charles Darwin was the one to really put that out there in print and get this idea really moving along in the scientific community. But it wasn't just Charles Darwin. Ernst Haeckel was himself a believer in human evolution from an ape-like creature, as well as Charles Darwin. And then Thomas Huxley was famously known as Darwin's bulldog, so to speak. And he was the one that not only pushed Darwin's ideas about evolution, but also his ideas about humans descending from apes. This whole idea of human evolution, which is now just taken for granted in the public schools and in the colleges. And so what is the current idea of human evolution? Well, according to evolutionists, they claim that humans evolved in Africa and then migrated out of Africa roughly about 120,000 to 240,000 years ago. Now, that none of them can ever agree on the exact date because this is all purely theoretical and purely speculative. But this is the general consensus. But there are some major problems with this idea, and these happen to be the locations of human fossils, what are now called archaic human fossils, but they're actually just humans that are outside of Africa. In fact, recently, They just dated some Homo erectus, or archaic human fossils, in China at 1.8 million years ago. Now, I don't buy into any of these dates, but I'm just showing you the inconsistency of these evolutionary concepts that are being pushed out there as, scientific fact, so to speak. Well, that's what are humans doing almost 2 million years ago outside of Africa? They supposedly evolved in Africa. But it's not just that. They've also dated archaic human fossils in Southeastern Asia, in Southeastern Asia islands at almost the same date, 1.7 million years ago. And so what's really interesting is this guy named Bruce R. Fenton. He's not necessarily a scientist, but he's a science writer. And so a lot of these scientific journalists, they're more astute really than a lot of scientists are, because they read everything all these scientists are publishing. And Bruce R. Fenton said, hey, if you got all these human fossils outside of Africa, maybe humans evolved outside of Africa and then moved into Africa instead of out of Africa. So he published this book, Into Africa, The Theory of Human Evolution. And so you can see the fossil record right off the bat isn't supporting this nonsense of out of Africa. But why do evolutionists even claim out of Africa as a hypothesis of where what they call anatomically modern humans came from? Well, it's because this is the only place in the world where you find chimpanzees. And we all evolved from chimps, right? So this is where we get the out of Africa hypothesis, as it were. Humans must have evolved there because that's where chimps are, despite the fossil evidence that says, hey, this is a bunch of nonsense. So anyways, that's where we get this whole out of Africa idea from. It's all rooted in this we evolved from chimps nonsense. And so chimpanzee, I would say in recent scientific history, has been chosen as the closest living ancestor to humans. I'm sure all of you have heard that at some point, right? We evolved from chimps, not gorillas, not orangutans, but chimps. Those are our closest living ancestors, supposedly. And so this is the standard, what we call evolutionary tree, or in scientific circles, we call it a phylogeny for human evolution. A lot of people laugh because they see John Wayne up there, but that's the pinnacle of evolution, right? Is John. Maybe I should put Clint Eastwood up there, Dirty Harry or something, right? So anyways, so according to evolutionists, we're most closely related to chimpanzees, then gorillas, then orangutans, and there is rhesus macaque, which is a monkey, as what we call an outgroup, so to speak. However, evolutionists never can agree amongst themselves hardly on anything other than that we evolved from some ape-like creature. There was a paper published not all that long ago. Let's see what the date here on this is. I've got my contacts in. One is close up, one is distance. I can't ever read stuff. What's the date? Yeah, 2009. Sorry about that. So this paper was put out in 2009, and it was called The Evolution of the Second Orangutan, Phylogeny and Biogeography of Hominid Origins. And in this paper, they said, hey, we're not more closely related to chimps because we share more anatomical features with orangutans. But the fact of the matter is that in my educated opinion, I believe all of the great apes are distinct created kinds. There's the orangutan kind, there's the chimpanzee kind, and there's the gorilla kind. We share some features with, anatomically speaking, with chimps, some with orangutans, some with gorillas, but there's no clear evolutionary pattern because humans were created in the image of God, and we are, you could say, a created kind. But anyways, these are all distinct created kinds, and that's why there's no clear evolutionary pattern here. And in fact, when I first began looking into this whole issue of how closely humans were related to the various apes, I began looking at different protein sequences. And so proteins are chains of amino acids, and so you can actually look at the sequence of amino acids and line them up using software, and it will create these trees showing how closely related these amino acid sequences are to each other. And so this is just one protein that I did, alpha motif protein 11. And in this case, humans are most closely related to gorillas. And then who do you see third down the line there? A rhesus macaque, a monkey. That shouldn't even be up there at all. And then next, you see an orangutan and then a chimp. And here's another protein sequence that I did, ACAP3. And once again, there's no clear evolutionary pattern here whatsoever. There's humans at the top, then right below that, there's a gorilla again, there's a rhesus macaque where it shouldn't be, then chimp, then orangutan. And so this is very common to see all these mixed up evolutionary trees when you look at these biological sequences that are very similar to each other, but this is the kind of thing you see. There are no clear evolutionary patterns, and I'm going to show you some evolutionary papers that were published that came up with the same data. In fact, here is one. When the gorilla genome was first sequenced, they created what we call a rough draft sequence for the gorilla genome. That was in 2012. And they said this in the paper, and it's the same thing I just showed you with these protein sequences, only this is with DNA. They said in 30% of the genome, gorilla is closer to human or chimpanzee than the latter are to each other. In other words, no clear evolutionary pattern here. And here was a paper that was published in 2007 where they actually looked at DNA sequences from the great apes and human, and they said this: for about 23% of our genome, we share no immediate genetic ancestry with our closest living relative, the chimpanzee. So the bottom line is, even before the chimpanzee genome was sequenced fully and the human genome, they still had a lot of data showing that there was no clear evolutionary relationship. So anyways, what about this whole idea of humans and chimpanzees being 98 to 99% similar in their DNA? How many of you have heard that at some point in watching TV or in college or whatever? You know, I've got kind of a funny story. When I first came to the Institute for Creation Research in January 2009, it was shortly after that, maybe 6 months after that or so, the president of ICR at the time, or I think we called him the CFO at the time, was Henry Morris III. And there were two biologists there. There was me, who had just gotten hired. Previously, I was working as a faculty member in genetics and biochemistry at Clemson University, and I ran a genome center there, and I sequenced the DNA of all kinds of creatures when I was there, but not humans and chimps. But anyway, and then there was another biologist that came from San Diego, because ICR had just moved their operations from San Diego to Dallas. And so me and this other biologist are sitting there, and this other guy now, he's no longer there. He went to work as a professor at a Christian university in Louisiana. But so we're sitting there, and Henry says to us, he said, we go into churches and we give talks, and we always get this question: are humans and chimps really 98 to 99% similar in their DNA? And I'm like, I didn't know what to say. I had never studied that, but the other guy said, and Henry said, and I want an answer in 6 months. And the other guy, I didn't say anything. I'm just totally silent. I just got in there, and I'm still trying to figure out, how do you work in creation science to begin with? I've been in academia my whole life, And the other guy said, Henry, it's going to take longer than 6 months. And he was absolutely right. This has been a journey, a literal journey for me, trying to figure out this human chimpanzee DNA similarity issue. It started in 2009, and I think I finally published my last paper in 2018. And at that point, I said, I've done all I can do. I'm just going to wait for the secular researchers to catch up with what I've found, and we'll talk about that. So anyway, so here was one of the first papers that was ever published comparing chimpanzee DNA to human DNA on a genome-wide basis. And so when the Chimpanzee Genome Project first got going, it was roughly about 2002. They had 3 million DNA sequences from chimpanzee, just randomly from all over the genome. And they were roughly about maybe 700 DNA letters long each, little snippets. And they tried to line those up onto the human genome to see how similar the genomes were, just kind of get a preliminary idea of what was going on. And they said this in the paper: about 2/3 could be unambiguously aligned to DNA sequences in humans. In other words, only 2/3 of those DNA snippets could they even get to match onto human. Well, that should have told you something right there. They're not 98 to 99% similar. But anyway, so they published that paper in 2002, and of course that didn't make it out to the popular media, did it? The only thing that made it out to the popular media was all these DNA sequences that were protein coding genes they were finding in the 1980s and 1990s that they already knew were similar to humans. Protein coding genes that were involved in metabolism and these common cellular processes, where not only would a certain protein DNA sequence in a common metabolic process be similar between humans and chimps, but between humans and rabbits and humans and rats and mice and stuff. So that's what, anyways. So that was the first paper that gave us an inkling, hey, things are not that similar. Well, let's review a little, some of my history on this whole project. So I was tasked to begin looking into the human-chimpanzee DNA similarity issue, and I had no idea what I'd find, really. I had no biases. When I worked at Clemson, I worked in all different kinds of plants in, a lot of crops like soybeans and corn and rice and things like that. Worked in a lot of tree crops like peaches, worked with a lot of different crazy animals like oysters and shrimp, even insects like fire ants and ticks. Yeah, we had fire ants and ticks running around in the lab, getting loose. I mean, there's all kinds of crazy stuff. So anyways, I had worked on a lot of different creatures, even flesh-eating bacteria at one point, which was questionable research. Anyways, but never had looked at the human-chimp DNA issue. And so I'm open-minded. I'm thinking, whatever I find, that's, I'll deal with that. So how do you start a research project? Well, you go and you dig up all of the research papers that have been published on that topic before you even begin doing any research. You got to see what's published. And so I took there was basically 6 major papers where they were claiming, or people were claiming from these papers, that the human and chimp genomes were 98 to 99% similar. And so I began looking at these 6 different papers, and actually Jerry Bergman kind of helped me out a little bit on this. He kind of got me going on it, and then I kind of took it over from there. And so we published a paper together, and I titled the paper, and it was published in the Journal of Creation in 2012. And the name of the paper was Genomic Monkey Business, Estimates of Nearly Identical Human-Chimp DNA Similarity Reevaluated Using Omitted Data. Well, the one thing I discovered right off the bat was reading these papers in detail, reading the materials and methods to see what in the world they even did, was they were literally only comparing DNA sequences between humans and chimps that were highly similar, that they knew previously were highly similar. They were throwing out everything else that was not similar. But there were a few papers where I factored in the DNA they threw out to figure out what the similarity was. And I was coming up with DNA similarities, depending on the paper, anywhere between 66 to 87% similarity between chimps compared to humans when I factored that data back in. And so what do we call that? used to have something on there that said cherry picking. So anyways, we call that cherry picking. So anyways, actually I think that's coming up in a slide here in a little bit. So at that point, the thing in my mind is, yeah, there's a bunch of monkey business going on here because they're cherry picking the data for the most part. They're just comparing what's highly similar, everything else that's not convenient for their evolutionary paradigm, they're throwing out. And yes, the algorithms that we have cannot compare stuff that's too dissimilar. Yes, there is that thing as well, but there's also this paradigm involved as well that causes people to toss out data. Well, let's do a little review on the human genome and the chimp genome. So in 2001, there were 2 drafts of the human genome that were published. So one was in the public sector, and actually Francis Collins was the guy that headed that up. How many of you know who he is? Okay, he's kind of a rascal. So anyways, he, at that time, this was young, early on in his career. You can see he looks a lot younger there than he does now. This is the guy that started Biologos. And so Biologos is essentially a theistic evolutionary organization where they promote human evolution. You might as well be on the Berkeley University website in California because there's no difference between what they promote compared to Biologos. So anyways, but Francis Collins headed that up. He actually didn't do anything in the lab himself. He was just kind of the guy heading it up. And there was another guy who headed up the private sector, and that was Craig Venter, and he was quite a character himself. And so they agreed to basically publish their versions of the human genome, their initial drafts, at the same time in 2001, and there they are shaking hands. And so anyways, so the first draft of the human genome, 2 different versions of it, was published in 2001. And they had billions of dollars to basically sequence the human genome. Then comes the idea, well, hey, let's get to the bottom of this human evolution thing. Let's sequence the chimp genome. And the first draft of the chimp genome was not published until 2005, although they started the project in 2002. But they did not have that much money to publish, to sequence the chimp genome like they did for humans, because they wanted to sequence the human genome because, you know, we want to get to the bottom of, well, why do we have, you know, heart disease and cancer and diabetes and all these things. So there was a very strong incentive to dump tons of money, especially from the National Institutes of Health or the NIH, into the human genome, but not that much money to sequence chimp. And I'll show you why that's important here in a few more slides. So anyways, the first version of the chimpanzee genome was published in 2005. And they were actually claiming a 98.5 DNA similarity between humans and chimps, but those were just for the highly similar regions. And so I actually began digging the data out of the paper, and it turns out that the alignments between the chimp DNA compared to human they were using were not 98 to 99% similar. They were 95% similar. And on top of that, they only sequenced 2.4 gigabases, 2.4 billion bases of the chimp genome. And so I just divided that by the known sequence of the human genome at that time, where they had sequenced 2.85 gigabases. And I came up with an 81% DNA similarity, just using basic math. And so at this point, I'm like, So how have evolutionists achieved these estimates of high similarity? They've omitted the non-similar regions of the genome. They've not included those in their figures, in their analyses. They've only compared just the highly similar regions or segments of the genomes. And as I said before, I call this cherry picking the data. And so that's exactly what has been going on there. So let's talk a little bit about DNA sequencing. What does that mean? Does that mean time's up? No. There you go. I'll stand up. Yeah, that was great. Yes, there you go. So anyways, contrary to popular opinion, DNA sequencing is not like Star Trek, where you go to some planet, you see some weird creature, you get a sample, you stick it in the computer, out pops this genome. It is not like that. Trust me. So let's talk about the Human Genome Sequencing Project. They had billions of dollars to do this. They had labs all over the United States, in Japan, in Europe. So it was this international effort. And every lab, say in the United States and in Europe and in Japan, they all took their own regions of the genome or certain parts of certain chromosomes, and they were assigned those. And they literally did by what we would call, and I'm not going to get into the technology here because would really be getting in the weeds. But anyways, let's just compare a genome to a set of encyclopedias to make things simple here. Each encyclopedia in that set would be a chromosome. And so with the human genome, they literally did a page-by-page sequencing strategy. Page by page by page by page. Very methodical, lots of money to pour into this. And so they got a very accurate, for the most part, for the first draft of the human genome, got a fairly accurate version, rough draft of the human genome by this methodical strategy. Well, let's talk about the chimp genome and how they went about that. And that was what we call an all-at-once sequencing strategy. In the genome business, which I was in for a long time, we called it shotgun sequencing. Shotgun sequencing. So let me tell you why we called it that. Think of taking an entire set of encyclopedias and to figure out what they all said, instead of reading them page by page, you throw the entire set into a paper shredder and shred everything up all at once. So you can understand why we're calling it shotgun sequencing. You shred every encyclopedia up. You got this massive pile of snippets of letters. So how do you put them all together? So you have to decipher the words on each snippet, which would be a DNA sequence, roughly about 700 to 100 DNA letters long, based on the technology at the time that we called Sanger-style sequencing. You look for overlaps between sequences, and then you can put certain groups of sequences together based on the overlaps where the letters match up. And then they took all those, what they called contiguous sections of DNA that they got from chimp, where they were to line up the letters on them, and they lined those up onto the human genome as a scaffold or as a reference sequence. Why is that? Because we descended from chimps, right? We're so similar to chimps, 98 to 99% similar. Hey, let's just throw this stuff, all these chimp sequences, on the human genome. And so what would be a good example? So how many of you like putting together jigsaw puzzles? So how do you do that? Well, you look at the picture on the box, or maybe there's a little poster that comes in there, right? And so with the chimpanzee genome, the picture on the box was human. So in other words, they put together the entire chimp genome based on the picture on the box, which was the human genome. They lined it up that way. So here you have all these clusters of chimpanzee DNA sequences that they were able to line up based on overlaps in these contiguous little chunks, and we call those contigs for short, and they line those up onto the human genome based on sequence matches. And so basically is what they had was all these sections of chimpanzee DNA sequence aligned onto the human genome with all these gaps in between them, and they didn't know what in the world was in those gaps, or if these chunks of chimp DNA sequence were even in the right places or in the right order. They just hung them up on the human genome where they could match them. And so what's really interesting is in 2018, the DNA sequencing technology actually improved substantially, and we now call it long-read DNA sequencing technology. So now, by 2018, they had the capacity to not create 700 base or 700 DNA letter long snippets, but snippets that were 30,000 to 50,000 bases long. And so when that technology improved, they were then able to say, hey, they were able to basically line those up one with another without using the human genome as a scaffold. Does that make sense? And they called that a de novo assembly. But look what they said in their paper that they published, and they basically resequenced the chimpanzee genome, the gorilla genome, and the orangutan genome, and they said this in the paper. The higher quality human genome assemblies have often been used to guide the final stages of non-human genome projects, meaning ape genomes, including the order and orientation of sequence contigs, and perhaps more importantly, the annotation of genes. And then they said this bias has effectively humanized other ape genome assemblies. So they actually admitted the previous versions of the ape genomes, and they did the same thing with gorilla and orangutan, sequenced them shotgun style and aligned them all up based on the human genome. They literally admitted these ape genomes have been humanized. And I'd like to make one comment here on the annotation of genes. One thing that I discovered in my research, that this big lab in England that was basically the central hub for putting all the DNA sequences together for the Chimp Genome Project, they were literally placing human genes electronically into the chimpanzee genome that didn't exist because they thought they should be there. I mean, I was blown away. And I was literally reading their methodologies they were using to annotate and put together the chimp genome, sticking human genes in there that didn't exist in chimp because they thought they should be there based on evolution. So I think that's what they meant by that, but they didn't say it like I just said it. They just kind of inferred it there. So anyways, but that's not the only problem, is humanizing these ape genomes based on the human genome. We have another problem. Several papers, one was published in 2011, one in 2016, where these scientists were going back into these public databases for basically everything but apes. They were looking at all kinds of bacteria, fish, mice, all these genomes that had been sequenced, and they were finding that some genome databases had up to 10% human DNA in them as contamination. Up to 10%. In fact, they sequenced the zebrafish genome, which was like the model fish genome. They sequenced the zebrafish genome and published it, and it turned out that 3 million bases or 3 million DNA letters on zebrafish chromosome number 3 was entirely human, totally human. That's how bad the human DNA contamination problem was. And they did another paper in 2016 and found out that the problem was even worse than they thought in 2011. In fact, there were bacterial genomes that were sequenced and published with human DNA in them, masquerading as bacterial genes. I mean, it's ridiculous. And in fact, human and bacterial gene structure is totally different. How do you, how do you stick human DNA in a bacterial genome and not notice it? Anyways, so how does human DNA get into the DNA sequencing reactions? Well, I ran a genome lab. I know what my people did in the lab. And people are coughing, they're sneezing, they're sitting there on their benches doing their reactions, they're talking to each other and carrying on and telling jokes, and you know what people do. And so there's DNA floating around in the atmosphere. In fact, in this room, there's human DNA literally floating around in the atmosphere, going through the HVAC system, and it's just everywhere. And I mean, not huge amounts of it, but there's enough of it. If you're running a DNA lab, it's going to contaminate stuff. And you're gonna sequence human DNA if your intention is to sequence some other creature. So, anyways, at this point, you can see what a can of worms this is that I'm getting into. Like when that one guy said it'll take longer than 6 months, it sure did, because I'm hitting all these caveats and all this stuff that's just like, wow, how do I deal with this? So I've got some questions at this point. Is the chimpanzee genome really accurately assembled or put together if they're using the human genome as a scaffold or a reference? Another question I had is, how much human DNA contamination is a problem in the chimp genome? Because they didn't test these ape genomes for human DNA. I guess they thought it would be too difficult because they think we evolved from monkeys. But anyways, so I've got questions at this point. And so I began doing some studies, but not only that, I'm like wondering what is the actual DNA similarity between humans and chimpanzees, and how can we figure this out? And so at this point, I'm saying I literally have to download my own DNA sequence from these public databases. By the way, your tax dollars pay for all this. And so the way these federal grants work, whether it's the NIH or the NSF, it doesn't matter. Once you produce a batch of DNA sequence, you are required by these federal agencies to submit that DNA sequence to public databases. And the main one in the United States is called PubMed. And so you have to submit that there. And so it's totally free for anyone in the world to download it. In fact, I could even download a DNA sequence from these European databases or these Japanese databases. So I began downloading my own chimpanzee DNA sequence and began working with it. So let's talk a little bit about DNA similarity real quick so we can kind of get on the same page here. So here are two theoretical DNA sequences. One is human there on top, chimp on the bottom. You have a T on top, and it matches up with a T on the bottom with chimp. And so in this case, you have a 100% similarity. You have an exact match for every letter there. And so T, A, G, and C's are these letters, which stands for the nucleotides in DNA. Well, here we have a mismatch for one DNA letter or one base. So in chimp, you have an A there in red, and human C, and because there's 28 bases there, so 27 out of 28 match, that's 96% similarity there. But it's not really that simple. In fact, now we run into the question of what gets included and what gets thrown out, because we don't have these simple mismatches. And we call those simple mismatches substitutions, which is kind of clothed in evolutionary lingo. But anyways, we have these substitutions, which are just single DNA letter mismatches. But the fact of the matter is we also have these gaps. So there you can see a string of letters in red there for human that's absent in chimp, that we just represent by dashes there in the chimp sequence. Does that make sense? We call that a gap. And so these gaps can be maybe a single DNA letter big, or they can be thousands of DNA letters big. So these gaps can be really small or really big. And when the gaps get really big, then the algorithm quits matching. It can't jump across that gap and keep matching. And so this brings up the point, what gets included in a DNA alignment and what gets thrown out because the gap is too big. And so this whole paradigm of DNA sequence similarity can get quite complicated, as you can see. And so we call these gaps indels, which stands for insertions or deletions. And once again, that's kind of clothed in evolutionary lingo there, but they're called indels. And so if we just count the substitutions here, which are just single DNA letter differences, we get 46 out of 48, or a 98% similarity. But if we include those indels for this theoretical alignment, we get an 81%. So you can see how evolutionists can play these games, because they essentially really only like working with the substitutions, because then they can run their theoretical models of human evolution where they claim we evolved from a common ancestor with chimps 6 to 8 million years ago. They don't use the sequences that have gaps in them. And so there's a lot of caveats to this whole paradigm. So anyways, I did a study I did a number of studies before this, but this is one of the most important ones I did. This was 2016, where I analyzed 101 chimpanzee DNA sequence data sets, and these were all the ones that were available at the time. There was 101 chimpanzee data sets. And then I sampled at random, and of course I used a random number generator, writing my own programs to do this. Sampled 25,000 DNA sequences at random from each of these databases, 101 databases or data sets, with a total, and I ended up with a total of 2.5 million sequences. And then I queried those against the human genome, and these were on average about 700 bases long. And so what I did is I used an algorithm called BLAST-N, and then I matched these chimpanzee DNA snippets onto the human genome. And when you do that, you get multiple hits all over the genome just because of small sections of DNA similarity. But I would take the top or the best hit and then record that, the best hit. And so, because the nice thing about this early DNA sequencing data is that not only did you get the DNA sequence from the database, but you also got metadata with that DNA sequence. So the metadata would tell you the date the DNA sequence was produced. It would tell you the location it was produced. It would give you the quality values on the electropherogram of the entire read for that DNA sequence. And it gave all this metadata. And so I dumped all of this data into what we call a relational database. And then I was able to connect DNA sequence similarity with the date that sequence was produced, which is very important. Now, if you look at these DNA sequences, there is DNA similarity on the y-axis. On the x-axis is the year that DNA sequence was produced. What kind of a trend do you see? The DNA sequences are getting less similar over time. Wonder what's going on there. Well, it turns out between 2002 and 2004, that was the data that went into that 2005 paper on the rough draft of the chimp genome. Well, between 2005 and 2011, the DNA sequence became less similar. And so this is just the basic numbers here. 2002 to 2004, the DNA sequence, the chimp DNA sequence was on average 92% similar to human for these top hits. Between 2005 and 2011, it was 85% similar. So based on all this contamination stuff I just told you, what do you think was going on in the first half of the Chimp Genome Project? Well, it was full of human DNA contamination, obviously. And so now we've got, like I said, this can of worms just gets more and more crazy the further I get into this. So finally, when the DNA sequencing technology improved, I then downloaded all of that DNA, not all of that DNA sequence, but 18,000 chunks of that high quality, what we call long read DNA sequence, which was void of human DNA contamination, praise God. And on average, those chunks of chimp DNA were 30,000 bases long. Now, those are pretty big chunks. And then I matched those on to the human genome. So this is good quality DNA, finally, in 2018. And that was actually from that paper I just showed you where they admitted previous versions were humanized. So I matched those onto human. The results were really crazy. Only 10,000 bases of each 30,000 base chunk on average would even match onto human. Only a third of that 30,000 base chunk could even match onto human before the algorithm hit a gap that was so big it couldn't continue matching. Of those chunks, 10,000 bases that did match onto human, the average similarity or identity was 84% similar. That was just what I could match, 84%. That didn't include all the stuff that wouldn't match. At this point, I'm thinking, okay, I've used this really good quality DNA. I've done all I can do. Nothing more I can do at this point. I just need to wait and let these secular labs come up with more data. And you can imagine the flack I took for publishing this stuff. 84% Tomkins, you're crazy, you're nuts, that's ridiculous, blah, blah. And then it just all these armchair YouTubers were putting up videos, and I'm just getting assaulted. But instead of playing whack-a-mole and putting up my own video and spending my life, responding to YouTube videos, I just decided to wait. Anyways, I need to mention this, because that same year that I published that paper with an 84% similarity, Richard Buggs, who was somewhat of a closet creationist, I probably shouldn't have said that, but anyways, he's a world-famous evolutionist at the University of London, Queen Mary. You better cut that out of this. Anyways, it seems like people in Europe just get away with more of this stuff. I don't know how they do it, because Europe is in some ways is worse than the United States, but yet you got guys like this. Anyway, so Richard Buggs is a world-famous evolutionist, actually, and he is actually the editor of a journal called Nature Ecology. And so anyways, so Richard Buggs did his own analysis, and he actually downloaded data from a genome browser, and it had been lined up onto the human genome. He did his own analysis using a completely different approach than me, a completely different approach. And he posted this on his blog, and he posted it on the BioLogos website. So I guess Richard really isn't in hiding necessarily, because he was going after all these BioLogos guys. Anyways, he posted that roughly shortly after I published my data. And he said this: the percentage of nucleotides in the human genome that had 1-to-1 exact matches in the chimpanzee genome was 84%. My same exact numbers. And so I should backpedal here. So at this point, I'm saying this is all I can do. Richard Buggs has verified what I did anyways. What else can I do at this point? So anyways, 7 years later, just last year in April, a paper was published in Nature, which is like one of the top scientific journals in the world, and this huge genome center in Seattle. It's world famous. They were the ones that did that 2018 paper where I was able to get that DNA sequence and do my own research from. They published a paper last year, and they resequenced the chimpanzee genome, the gorilla genome, and the orangutan genome using multiple individuals. And they literally did what was called a telomere to telomere genome sequence. In other words, they literally sequenced the genomes from end to end, even going through those areas which were really previously really difficult to sequence through for a number of reasons. And so highly accurate. They said that, and I should mention this, they published a 17-page paper in Nature. This was it. They said nothing about the overall genome similarity in the paper. In fact, they doubled down on this ridiculous evolutionary meme of 98.5% DNA similarity. And I'm thinking, this is really weird. And then they said, here's a link to the supplemental material. The supplemental material was 171 pages long. It was a literal book. And so, I really looked forward to going through that. But I had to. So anyway, I'm diving into this supplemental material, and I hit these tables in there, and this is what they said, and this is what was in there. They did what was called a haploid genome comparison, so a single genome equivalent. Now, you have two genomes in every cell in your body. One is from your mother, one is from your father. 3 billion bases from your mother, 3 billion bases from your father. So one of those genomes would be called a haploid or 1N equivalent. So they created a chimp genome that was a 1N equivalent compared that to human, and they said 85% DNA similarity or identity. And that did not include the stuff that was present in chimp, absent in human, present in human, absent in chimp, which is significant. And then they compared the diploid chimp genome against the human genome and came up, and they stated this in this supplementary material in this table, 84%. I had waited 7 years for this. Finally, you know. And I will say a lot of these evolutionists have gone silent. A few have just doubled down and just become insane in what they're saying. But anyways, it's pretty much settled. Maximum similarity between humans and chimps, 84%, which I had figured out 7 years before, but no one believed me, except Richard Bugs. So, why is that important anyways? What is the evolutionary problem with 84%? Why are all these evolutionists so up in arms and so freaked out? Or not saying anything, I should say, at this point. Because theoretical evolutionists need a 98.5 to 99% genome similarity to explain human evolution with their theoretical statistical models with known mutation rates in humans and known mutation rates in chimps, which are about the same, in 3 to 6 million years time since a common ancestor with chimps. So their speculative, theoretical, deep time calibrated models won't work with 84%. It totally boots evolution out the door, or human evolution. So anyways, yes, thank you. So anyways, I talked about this earlier today, but the fact of the matter is the human genome is not evolving, it is devolving. And John Sanford, through his theoretical modeling of the human genome, not only with himself, but other scientists, and John Sanford was a very capable guy. He was a Cornell University professor for many years and was actually an evolutionist for much of his career, and became a creationist late in life. Doesn't happen very often, but he was confronted with the data for creation and couldn't deny it, and got born again and saved and became a creationist. So, well, this makes sense because human lifespans, plummeted after the flood. We went through a genetic bottleneck at the flood, so the world was repopulated from Noah's 3 sons and their wives. And I'm not saying that this loss of human longevity was solely due to the buildup of mutations in the genome. There was environmental factors playing into that as well, but it was connected. And I talked about this study a little earlier. Now, this is solid, hardcore empirical data here, done by secular, conventional evolutionary scientists. Actually, these guys were in the biomedical community because they were actually trying to identify all of the mutations in human protein coding genes that were connected with various human diseases, like diabetes, heart disease, cancer, and birth defects, and things like these. And so they were looking at what they called rare variants, not common variants. Common variants just contribute to all the variability we see among humans, so in human skin color and height and these things like that. Now, these are called rare variants, and over 80% of them were connected to one form of human disease or another. So these mutations were doing nothing good, nothing good whatsoever. And they actually decided to create a molecular clock from this data, but they did not calibrate their models with deep evolutionary time, because that's typically how evolutionists work. They calibrate their molecular clocks with deep evolutionary time, which doesn't work because their molecular clocks that are calibrated that way never match up with paleontology, their own paleontology. And so, but anyways, so they calibrated the molecular clock from these studies, and there was 2 of them done. One was in North America, thousands of people across North America. They sequenced the protein coding regions of the of the human genes in North America and Europe, thousands and thousands of people. And they calibrated their molecular clocks with how fast populations grow, how many children people have, how fast people reproduce, empirical demographic data. And they literally said that this rare variation, these mutations, only went back 5,000 to 10,000 years ago. Now, this is the kind of molecular clock data that's real. And it also matches up completely with the scriptures. Because the Bible, the chronologies and genealogies that we have in the Bible basically tell us that from the present time back, or from the time of Christ back, is 4,000 years. And from now back would be 6,000 years. So this completely matches up. I just freaked out when I saw these two studies, and they were both published at the same time, so they must have agreed to publish at the same time together. One was in Nature and one was in Science, the two top journals in the world. So anyways, what about this whole idea of human evolution? Anyways, how am I doing on time here? Well, I guess we're running out of time. Okay, well, I'll go through this pretty quick. So, how many of you seen this so-called progression of human evolution? Well, the fact of the matter is all that we really have in the fossil record is apes and humans. And so what's really interesting is that they have found data corresponding to humans going back to supposedly the very root of evolution, if you want to accept these millions of years. So they have found ape fossils going back, the so-called ancestor ape fossils to humans, supposedly going back to about 4.5 million years. However, they have found anatomically modern human bones, including archaic human bones, back to a little over 4 million years. They have also found human tools, artifacts, and footprints going back even before the supposed first proto-human ape creature even existed. So we have major problems here with the real data and the theory. One of the biggest problems is the Laetoli footprints, which were discovered in 1978 in Laetoli, Tanzania, dated at 3.5 million years ago. So who made these footprints? Well, there's a human foot and there is a chimpanzee foot, or an ape-like foot there. And you can see there's a complete difference between the two, because ape hands and feet, or at least for chimpanzees anyways, are made to climb around in trees. And then when chimps are on the ground, they have locking wrists so they can knuckle walk on all fours. There's the Laetoli footprint. It completely matches up with a human footprint. And in fact, Russell Tuttle, who is an evolutionist, in this article, The Pattern of Little Feet, the American Journal of Physical Anthropology, said that they were indistinguishable from those of habitually barefoot Homo sapiens. But it gets even worse, because we actually have hard data. This is Olduvai hominid #1, an anatomically modern human skeleton that was discovered in 1913 in the same strata as Australopithecus fossils. And the guy that discovered this knew he would get kickback and pushback. He knew the evolutionists would say, oh, that was a human that was just buried there recently. Well, he called in a geologist before he even completed what he was doing and had it authenticated that this individual was actually buried in situ. It was not a grave. And I think he was buried in the flood, in the receding phase of the flood. So anyway, so we have this. And so in other words, the first ancestor supposedly of humans, which was a completely ape-like creature, Australopithecus, dates back to the same ages as anatomically modern humans. So we have no human evolution going on here whatsoever. And so we have the Lucy fossil, which is an Australopithecus afarensis. I remember the guy that discovered this, Donald Johanson, came through Washington State University when I was right after I don't know if I'd gotten saved yet or not. I was right about that time. And he brought Lucy there and showed her off, and it was just a big deal. And Anyways, so Lucy is only about 40% complete. And in fact, the lower part of Lucy is kind of disputed as if she even belongs to the upper half, which I don't think it does. But the upper half is definitely 100% chimpanzee-like. And she was dated at 3 million years old. And it was claimed that she walked bipedally or walked upright. However, anatomical anthropologists came in after she had been discovered and began examining Lucy, and there was a lot of very interesting comments about her. So this is Eric Stockstad in an article, Hominid Ancestors May Have Knuckle Walked. He said, I walked over to the cabinet, pulled out Lucy, and shazam. She had the morphology that was classic for knuckle walkers. And then this, it was said in this article in Science News, I love the title of this article, Lucy on the Ground with Knuckles. For those of you young people, that was a play off of a Beatles song, so anyways. Australopithecus anamensis and A. afarensis, the latter represented by the famous skeleton Lucy, this is a quote from the article, known as Lucy, had wrists capable of locking the hands in place during knuckle walking. And here's another article, evidence that humans evolved from knuckle walking ancestors in Nature 2000. Regardless of the status of Lucy's knee joint, new evidence has come forth that Lucy has the morphology of a knuckle walker. On top of that, I have also analyzed, I probably should have included these articles, but it would have made things way too long. But articles have been published the past 15 years showing that Lucy's scapula and her shoulders were entirely designed by God, engineered by God to swing around in trees. In other words, she had an arboreal adapted anatomy. And I've also read articles on her skull and brain showing that she definitely had a chimp-like brain and skull. So, and the evolutionists agree with all this. So this is a paper that was published, I mentioned this paper earlier today, in the Royal Society, a very prestigious journal in Great Britain. And they said, I love the title of this, From Australopithecus to Homo, the Transition That Wasn't. Although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented. So what about Homo habilis, handyman? Well, this is now widely considered as an invalid taxon, a mixture of ape-like and human fossils, which seems to be a common problem with a lot of these constructions, especially with Lee Berger and his stuff. Anyways, so here was a quote from Milford Walpoff, the American Journal of Physical Anthropology. He said, if there weren't a Homo habilis, we would have to invent one. If that isn't the truth. So what about all these archaic traits, these so-called archaic traits? What about Neanderthals and these so-called traits? So now we're seeing pictures of Neanderthals like this, and they look pretty look pretty human to me, except for the ornery looks on their faces. I guess that makes them look like they're primitive, right? If they're all hacked off. They're probably hacked off because they're being called archaic. Anyways, so Eric Trinkus said this in Hard Times Among the Neanderthals in Natural History. Detailed comparison of Neanderthal skeletal remains with those of modern humans have shown there is nothing in Neanderthal anatomy that conclusively indicates locomotor, manipulative, intellectual, or linguistic abilities inferior to those of modern humans. And now the really hot thing is, if you want to really be a rock star in anthropology, you're out there sequencing Neanderthal DNA. And so this guy here, Svante Pablo, is at the Max Planck Institute in Germany, and there he is holding up a Neanderthal skull in front of his skull. And so he's quite the rock star in this field. He's been publishing on this for years. And so anyways, now we see papers popping up like this all the time. Humans bred with this mysterious species more than once, new study shows. And now we see even more stuff like this. Your Neanderthal DNA might actually be doing you some good. So there has actually been talk in the evolutionary or anthropological community, as it were, about cloning Neanderthal DNA brain genes and splicing them into our genome to make our brains bigger. I guess they weren't thinking along evolutionary lines there, were they? And so, yeah, so anyways, ancient humans may have been mothers to some Neanderthals earlier than we thought. And so the fact of the matter is now, and I just read a paper just a few weeks ago, how that Neanderthals now were readily interbreeding with modern humans. Now they're finding caves in Israel where with ritual burials, where some of the burials are Neanderthal, some are anatomically modern human, and some are all in between, indicating they were all having families together and having children together, and they were just humans. So anyway, they have even found genes for red hair in Neanderthals. And what about Homo erectus? I'm wrapping things up here real quick. So Homo erectus skull is very similar to Neanderthal skulls as far as the sloping forehead and the large brow ridge, only they're just smaller and more in line with modern humans as far as the cranial capacity. And so here is Turkana Boy. He had a skull that was considered to be archaic human because of the large brow ridge and the sloping forehead, but everything below the skull was anatomically modern human. Well, the fact of the matter is he was just human. And so Neanderthal and Homo erectus were just basically humans. And so what about these traits anyways? And so I showed this picture earlier today if you were here, but there's a dentist in South Africa that takes pictures, he's a creationist, he takes pictures of patients that have these alleged archaic traits. And so he took a picture of this guy, and look at the size of the brow ridge on this guy, and I can assure you he was alive and well. And here's some rugby players from Eastern Europe, and sloping foreheads and large brow ridges there. And this is one of my favorite pictures here. And this guy actually just takes great pride in being called a Neanderthal, actually. And he's a big guy, actually. He's very tall. But anyways, he was a professional boxer. He was undefeated except for one match he lost. Then he became a member of the Russian parliament, a very smart guy. And look at that, that sloping forehead, incredibly sloping, the large brow ridge. And the fact of the matter is you could go down to the local mall and sit there and see people with alleged archaic traits like that. And so what about this whole thing called the sagittal crest? And so this is a ridge that runs down the center of the head. Some people have this, some people don't. And there's the kung fu guy there, has got one right down the center of his head there. And then the guy up on the left in the suit is a world famous neurosurgeon. And I assure you, he's pretty smart. And of course, the guy on the right there, I mean, he pilots a star cruiser across the galaxies. I mean, he must be pretty smart too. So anyways, there's just a lot of variation in humans, but it is not evolution. It is just variation. And here's two tennis players. One's almost 7 feet tall. The other was 5'7", and this was actually a really close match. And so anyways, and so this is all very important, but why is it important? Why is the enemy so after humans with this human evolution stuff? It's like the pinnacle of the evolutionary paradigm, humans descending from apes or chimps. It's like that's like the main thing. And why is that? Well, it's because the devil is so mad because humans are created in the image of God. And so Genesis 1:27, so God created man in his own image. In the image of God, he created him, male and female, he created him. And this all boils down to the historical Adam. Now, this BioLogos organization, which Francis Collins helped start, believes that humans descended from apes. Like I said, you might as well be on the Berkeley University website. You'll get the same story. And some of the people that are associated with BioLogos believe that Adam never existed. There was never an Adam. Other people say, well, God, at the certain point in human evolution, God picked a couple of hominids out as Adam and Eve and made them representatives of the human race. And so but anyways, it's all absolute nonsense. Why is that? Because the gospel is basically based on two Adams. The first Adam, Adam and Eve, Adam who plunged us into death and corruption, who brought total depravity into the human race. How do you explain death, corruption, and the problem of evil in the world without him? And then we had the second Adam, Jesus Christ, who lived a perfect life, completely fulfilled every bit of the law, and then gave himself for our sins. And he is called the second Adam because he brought us redemption compared to what the first Adam brought us, which was corruption. And we actually have detailed chronologies from Christ. One is back to Abraham, the other is literally back to Adam. And so the Apostle Paul thought this was really important. And he said in Romans 5:12, therefore, just as through one man sin entered into the world, and death through sin, and thus death spread to all men because all sinned. And then 1 Corinthians 15:22, he brings it up again to a completely different church he's writing an epistle to. For as in Adam all die, even so in Christ all shall be made alive. Well, can you be saved and still believe in an older? Sure. But it doesn't help the gospel much, does it? It doesn't help it at all, actually. In fact, it doesn't help a lot of things. How do you explain, pain, disease, and suffering if you can't have a literal Genesis and a literal Adam? Very difficult to do. It really messes up the Bible. So anyways, I've got this book on chimps and humans, which I go into everything on this topic. So not only have I been looking at the DNA similarity between humans and chimps for past 15 years, I have also been looking at all the other so-called paradigms, the chromosome fusion, the pseudogene thing, literally everything I address in this book and totally debunk it. And then I have a book on the design and complexity of the cell, which shows the incredible design in cells that God created. The fossils still say no. I had a talk on that earlier today, and that's on the paleontology of the flood, and then Creation Basics and Beyond, which has a little bit of everything in it. And we have a Discovery Center in Dallas, which I would recommend seeing. It's great, really, for all ages of people. Even bringing skeptics to it is a great thing to do as well. And so we have a bi-monthly magazine that you can get for free. You can sign up for that, and you can actually scan that QR code in there to sign up for that. And I think we also have some sign-up sheets out there in the lobby as well. So that's it, and I apologize for going a little long. So.